Well, thanks so much. It's a real privilege to be here to speak to you all. I'm going to be, I guess, pivoting a bit to talk about things that we do to patients to evoke reactions on the background of risk genetics, focusing on T-cell mediated reactions. So my objectives today are really to give you an overview of how immunogenomics has given us an insight into evolutionary context of these reactions. What is the translational roadmap for prediction and prevention, and how might we use these to increase drug safety, and then how does this integrate into multi-omic approaches, increasing our knowledge as we get more information over time. And so the concept of immunogenetic evolution is really, when we're talking about largely about the major histocompatibility complex, has been evolving for over 400 million years from the time of the first vertebrates, essentially jawed fish. And these are experiments of nature where we've evolved basically to protect ourselves from pathogens over time as new onslaughts come on, like COVID and other things. And then the last 120 years have really been a very short time where we've made drugs to treat in patients. So the intentional scale up of the pharmacological revolution, which has led to adverse drug reactions of all sorts, but in terms of the immune ones, we can think of these drugs as being probes of our immune system. And so our HLA is co-evolved with us. I was really interested in Noah's talk because actually HLA is the one thing that actually tends to negatively predict. Similar HLA tends to negatively predict mate selection, so we tend to choose mates that are not HLA similar to us, and this actually has a reproductive advantage, and so the reproductive success is higher when HLA is not similar. And this also has an evolutionary basis, as there is something called the heterozygote advantage with HLA, where we can actually effectively present and better peptides and antigens across scale if we're heterozygous at the HLA level, and this has been shown with HIV and other pathogens quite nicely. And so HLA, as you all know, is the most polymorphic region in the human genome, but it also differs from many other genes in that it's codominantly inherited, meaning that we get one copy from mom, one copy from dad, and both are active. And so in terms of the genetics of HLA, we think of the most active regions that are actually involved in antigen presentation being class 1 or class 2. These are both polymorphic, but most of the polymorphism is involved with class 1, which is expressed on all nucleated cells, essentially, so all cells except for red blood cells. And these are involved in basically processing intracellular pathogens like viruses and parasites, and present these to CD8 T cells, which are the cytotoxic arm of the immune system responsible for a lot of the heavy-duty action, especially with regards to viruses and intracellular pathogens. And then we do see when things go wrong, of course, which they can, this is where we see these so-called experiments of nature, most commonly autoimmunity, and of course drugs hypersensitivity we consider to be a iatrogenic event or experiment of humankind. And so these types of experiments of nature that have been HLA class 1 restricted or HLA class 2 restricted in the setting of type 1 diabetes and others, or autoimmunity, but the HLA class 1 restricted experiments of humankind that we're seeing are largely related to drug hypersensitivity. I just wanted to give you a landscape of what drug reactions are, because I'm really focusing on these terrible immune reactions along this scale, which are the T-cell mediated reactions, but there are many other types of reactions, many of which, interestingly, can wane with time or do not stay with us over time. Penicillin allergy is an example of an IgE-mediated reaction, which tends to wane with time. We can show this with skin testing studies, but in terms of T-cell mediated reactions, these tend to stay with us over a lifetime, so it's a lifelong memory T-cell response. So what maintains this lifelong response to the drug? Because it's curious, we don't take the drug again, usually after it has caused a terrible reaction. So how come this reaction persists despite the absence of the drug? And we think that this is due to something called a heterologous immune response, which occurs amongst different pathogens. It occurs amongst viruses. It explains why when a new pathogen is introduced into an environment that there is often morbidity, but the mortality is fortunately usually controlled. And what happens in this hypothesis is what we call a two-hit hypothesis. And so early in life, there's a response to an intracellular pathogen, such as a virus, which is HLA class 1 restricted. And then the drug comes along years later in a HLA at-risk person, and that drug actually is able to alter a self-peptide to look like the viratope, and this creates a response that actually mimics the original response, but of course it's a mistake, and so it leads to some form of hypersensitivity reaction. And what we're still working on is identifying what actually are these antigens and how do they actually lead to these different phenotypes of drug hypersensitivity. And so the story of the human leukocyte antigen has really been one of disease protection and susceptibility, both across class 1 and class 2 alleles, but especially in terms of intracellular pathogens and drug hypersensitivity and certain forms of autoimmunity, class 1 HLA, and you can see that the discovery of these associations with drug hypersensitivity came very late to the piece, so after 2002. The timeline of this is shown, and you can see before this it really wasn't really understood from an immunopathogenesis standpoint how these reactions even occurred. So the genetic association was key in defining the immunopathogenesis and everything that followed. So the genetic association between in particular between HLA-B5701 and abacavir in 2002 really set the stage for everything that followed in terms of these understanding these reactions to be CD8 T cell dependent largely in HLA class 1 restricted, and this led, in the case of abacavir and 5701, it led to a randomized control study that actually tested the effectiveness of this allele to actually prevent a specific drug toxicity, which happened to be abacavir hypersensitivity, and showed that it essentially screening eliminated that toxicity, and this led into universal guideline-based screening. Unfortunately, we don't see abacavir hypersensitivity anymore in clinical practice, and a number of other drug associations, which I'll speak to, actually followed this. And so another key question is, how do drugs act as antigens, or at least small molecule drugs less than 1,000 kilodalton? How do they actually activate T cells? Because they're not immunogenic, they're not antigenic by themselves, and so they have to somehow alter a protein to be recognized by the immune system. And until recently, it was thought that all drugs haptenated or binded covalently to these proteins, but we think that with most of these, including abacavir, that there's non-covalent dose-dependent binding that actually leads to this alteration. So it's an irreversible binding or a reversible binding process. I wanted to show this slide to show a study that we did a few years ago, and this was led by Jason Carnes and Josh Denny, also involved Dan Roden, and in this study we were able to reproduce 25 years worth of discovery, largely autoimmune illnesses related to HLA class 1 and class 2 alleles, things like type 1 diabetes, ankylosing spondylitis, etc. Be able to reproduce all of those doing a HLA-PheWAS approach using HLA-imputed array data from our de-identified electronic health record and of about N equals 37,000. But the one thing that we couldn't do with this data is actually pick out drug reactions. And the reason for that is, one, they're rare, and two, the actual deep phenotyping of these reactions to find this in the EHR is actually not being perfected. This is something we're still actually working on. And so HLA class 1 restricted T-cell reactions, what are these? I talked a bit about abacavir hypersensitivity. There's also severe cutaneous reactions, and then there's organ-specific reactions like drug-induced liver injury. These are some examples of these. These are actually quite rare, as you can see, only affecting 1 to 6 per million per year in the case of Stevens-Johnson syndrome and toxic epidermal necrolysis. However, they have a high mortality. So for DRESS, the case mortality is up to 10%. For Stevens-Johnson syndrome, it can be 50% or even higher in elderly or immunocompromised. And for drug-induced liver injury, there's about a 10% mortality and about 1 to 2% go on to liver transplantation. And so many of these HLA class 1 alleles are strongly associated with cutaneous T-cell mediated reactions, and these are examples of drugs that, other than abacavir, which doesn't cause an actual DRESS or SJS, it's its own syndrome. These have been associated with largely DRESS or SJS, which are strongly class 1 HLA restricted, and you can see these are caused by all different HLA class 1 alleles, as you can see there. What they do differ by is the number needed to test to prevent one case, which is critical. This was critical that abacavir was an easy example because 55% of those that actually carried 5701 actually developed abacavir hypersensitivity, and the carriage rate in the population being treated at the time, which was largely European, was about 7%. So this meant that the number needed to test to prevent one case was only 13, whereas you can see for these other examples, this goes up significantly. This is something, these gaps that we don't completely understand on an immunopathogenetic basis, but it does actually affect how we actually think of these and use these in clinical practice. And similarly, drug-induced liver injury has been associated with many of these. Most of these are small molecule drugs, but green tea and catechins and polyphenols and many health products have also interestingly been associated with an HLA class 1 allele, which is B3501. So these implementation gaps, which from a safety standpoint, the most critical one is really the negative predictive gap, and that means that if you have an HLA allele that's been discovered in another country or geography, it doesn't mean that that's going to apply to your population, and you may miss cases if you treat it like abacavir and 5701 and say that we can just screen, we can give the drug to everyone that's negative and they won't get a reaction. That's not the case for the majority of these reactions, and that's what I'm going to focus on the next few slides. So this is a safety alert. As I've said, a single HLA risk allele does not identify all patients at risk. For most of these, HLA alleles have been evolutionarily determined to be geographically distributed under the pressure of pathogens and migration that occurred out of Africa millions of years ago or hundreds of thousands of years ago. There's heavily admixed populations in the USA and Europe, and these patients have diverse risk. And so this is an example, again, with abacavir, why this was an implementation success story, largely because of the low number needed to test to prevent one case. This could actually be done in a randomized control study that enrolled almost 2,000 patients in 6 months, and also because HIV providers at the time had very huge practices. When they started to implement this, they could reality test that this was working. All of a sudden, they weren't getting calls about patients having fever and rash, and so it just extinguished itself from clinical practice. On the positive predictive side, we know that HLA is necessary but not sufficient. So for abacavir, this number was 55%. For other drugs, which I'll talk to some, this is a much lower number. For the anti-staphylococcal agent vancomycin, it's 20%. For HLA A3201 and for allopurinol B5801. One thing that we did learn from abacavir was the actual mechanism, and it was elegant work. I was fortunate enough to be involved in the study on the top left there, but this was elegant work that actually, in 2 cases, solved the crystal structure of abacavir bound to a peptide and HLA B5701. through elegant studies that included a combination of peptide elution, peptide binding studies, it was found that abacavir actually binds within the F binding pocket of HLA-B5701, and it alters the type of peptide that actually binds to B5701, and the typical native peptide would be an aliphatic aromatic peptide, and it changed this to an aliphatic linear peptide like valine or leucine, and based on this, this creates a foreign response that can now be recognized by CD8 T cells. And so this was a paradigm shift in our understanding, and further understanding was increased by the fact that in a transgenic mouse model, the depletion of CD4 T cells actually seemed to facilitate maturation of dendritic cells and seemed to be a prerequisite to developing abacavir hypersensitivity. So it could be postulated that all patients actually, abacavir alters the F pocket in all patients that get it, but only some of them actually are immunologically predisposed. In addition, we had shown that of the 55% of those that develop it, B5701 is necessary but not sufficient, and this is shown here, where there was one patient that didn't have any of these other haplospecific or haplotypic markers, and this was also a caveat to saying that we could develop, TACMAN assays for some of these SNPs like HCP5. That would be a safety issue. So all of the screening alleles actually had to be HLA-B5701. And we showed actually that those that actually endoplasmic reticulum aminopeptidases trim peptides to the size of 8 to 10 mers that can be actually recognized and presented by antigen-presenting cells and HLA-expressed antigen-presenting cells, and this was also overrepresented in those that actually had abacavir hypersensitivity. They were less likely to have a hypoactive allotype. And so global representation of HLA alleles related to drug hypersensitivity, this shows that there's different HLA alleles that are represented geographically, again, based on evolutionary selection. And you can see that this is actually significantly different. So the ones that are prevalent in Southeast Asia are not those that we see frequently in Europe or Africa and onwards. And one interesting thing is that India has a very broad representation of HLA, and India has one of the highest rates of SJST and DRESS and drug-induced liver injury in the world, and we think it's because of the representation of all of those risk alleles. So I'm just going to present a case study. This was a young man that we saw with trimethoprim sulfamethoxazole. It's a common antibiotic, sulfonamide antibiotic, and he had developed this just being treated for a skin reaction, and this is what he looks like at presentation, and then at day 20 when he had started, when he had complete healing. And we studied this in the HLA basis for trimethoprim sulfamethoxazole, but I wanted to give you the background that the studies that had come out of Southeast Asia had shown it to be associated with specific HLA class 1 alleles, namely B1301, which is the strongest one, and then several others on systematic review. And when we looked at this in the US population, we found that we did not find B1301, but we found B5403, which shares peptide binding specificities with B1301. And we also didn't find the second risk allele, but we found B3801, which also shares peptide binding specificities. So we think what's going on here as part of this evolutionary context and story is that HLA has shared peptide binding specificities, so we have 28,000 HLA class 1 alleles, and for efficiency purposes, they're overlapping in terms of the peptides that they present. So there may be 28,000 alleles, but only 12 supertypes, and it's these supertypes that actually share these antigenic peptides and present these overlapping sets, and this has enabled convergent evolution, but it also means that in terms of drug hypersensitivity, you're going to get different HLA alleles that have these shared properties in different populations. And so this young man had B4402. And this is just a global map of cotrimoxazole based on that story. We've also found that B5801, which is the prevalent allele found in Southeast Asia that explains allopurinol scar, that a high proportion of our populations did not have this. In fact, it was quite striking that 45% of US blacks did not carry B5801, but they carried an HLA-A allele associated with both allopurinol address and SJS-TN, and this work's coming out next week. This is a caveat also about lamotrigine toxicity and lamotrigine epidermal necrolysis, that many of the commercial genetic tests that are available actually do HLA B1502 and HLA A3101, which are not associated with lamotrigine TEN. And this young woman was told when she had genetic testing that she would be unlikely to get it. And this is what the report looks like. It says use it as directed. It has a green bar. But what this use as directed means is use lamotrigine with typical caution because it's not associated with these HLA alleles. And we're showing the same thing with lamotrigine. Just beyond prediction and prevention, we have shown an association with an antibiotic with HLA A3201 and think this will be more useful to be used for diagnosis and preemptively because vancomycin is often co-dosed with antibiotics and it becomes an issue of which drug has actually caused the reaction. And you also have a time window to do screening after you've started an antibiotic for life-threatening infection. And I just wanted to end by talking about some of the work that we've done at the site of tissue damage. This has been years really of biobanking these samples to actually look at the immunopathogenesis at the site of SJS and dress, blister fluid, and skin. In SJS, we found that the T cells that we find in the blister fluid in the skin that we don't find in the peripheral blood. So that was actually a huge clue that if we were going to study immunopathogenesis and look at these dominant TCRs in the blister fluid, that we would have to get them from the blister fluid and not the peripheral blood. And using this, we use single-cell techniques to create an atlas of cutaneous reactions and also learn a lot about SJS-TN in terms of we found in SJS-TN blister fluid single-cell studies that there were similar signatures across all drugs and HLAs, although they all had private TCR clonotypes, but these antigen-presenting cells, the keratinocytes in the skin, increase of presentation, increase the expression of class 1 HLA, and then presenting antigens to CD8 T cells. So they're actually doing themselves in. It's like sort of a completely catastrophic vicious circle, if you think of it that way. We found that certain pathways were upregulated, which opens a pathway to thinking about how to step in early with treatment because TNF-alpha antagonists and JAK inhibitors are available off the shelf. And then we've done more work looking at the programming at a tissue level and found that these T cells that are at the in the blister fluid in SJS. When we look at skin and spatial studies, we see that they are at the dermal-epidermal junction and expressing residency markers. And interestingly, they also express long non-coding RNAs, suggesting that there's epigenetic regulation of these T cells at the dermal-epidermal junction. All of this, as I've mentioned, lends itself to thinking about targeted therapies, which there's currently no evidence-based therapy for any of these severe reactions. So just to close, these T-cell mediated reactions have strong HLA associations that have taught us a lot about the immunopathogenesis, so the genetics came first and drove our understanding. There's lots of gaps to consider, and populations have to be considered separately, although the shared HLA supertypes provide a context and biological explanation for actually how to manage these, and maybe to think about doing panels for risk assessment rather than individual HLA alleles. Current treatment approaches, as I've mentioned, lack high-level evidence, but these single-cell studies and us understanding a lot more about the immunopathogenesis also open areas for development of prognostic and diagnostic biological markers, as well as new treatment targets. So I'd just like to thank my group at VUMC, at the Center for Drug Safety and Immunology, all of the patients, families, foundations in particular, and funders, and then lab members and collaborators. And thanks so much for your